All right, our final speaker, last but not least, our president of SDC, Dale Usner. He'll be speaking on adaptive design, practical considerations, and execution. I've had the pleasure of working with Dale over the past 9 years, first as a client, and over the last 6 years as a colleague. We've probably collectively flown a couple of million miles together in those past 6 years. But one of the many great things about working with Dale is he's able to help people understand the importance, impact, and implications of items from a statistical perspective, regardless of that individual's role within the study team. Prior to joining SDC, Dale's experience included leading stats and data management teams at sponsor companies, AVI Biopharma, Cymogenetics, and Bausch & Lomb. Also, large CRO experience with PPD prior to that, as well as statistician here locally at NERI. Dale has an extensive array of therapeutic experience, probably easier to list areas that he hasn't touched at this point than what he has, and a lot of extensive experience as well interacting with regulatory agencies on behalf of clients, and prior to being at SDC on behalf of his sponsor companies. So with that, I will turn it over to Dale.
Thank you very much again. Thank you, Jim. Thank you, Ken and Joe. Everybody, what I'm going to be going through now is sort of broadening, hopefully, people's minds on what adaptive designs are outside of increasing sample size or stopping for futility, but doing some other adjustments that probably will add to the complexity that Ken was mentioning, at least on the short run, hopefully reducing the complexity in the long run. And all right, so the key two references I used for this were the two guidance documents from the FDA, one from the Drugs Division and one from the Device Division. The Device Division is final, the Drugs Division is draft for 7 years. They were pretty similar, so I think they used each other. My agenda, I'll be going over the definition, some of which was already discussed, motivations, disadvantages of adaptive design, what you're able to modify or what's currently being discussed as possibilities of modifications in trials, interim data and what you'll do with it, candidate studies, so where is it good to possibly have an adaptive design versus not, examples, and then questions and answers. And I don't stand well behind this, so if I'm in front of somebody, tell me to move. Definition of an adaptive design clinical trial, prospectively planned. You've heard that before, you're hearing it again for a reason. It must be prospectively planned so that you are able to essentially keep the bottom bullet designed to maintain study integrity and scientific validity. It's an opportunity to modify one or more aspects of the study design, study conduct, hypotheses, or analyses. You can build an adaptive design to change your primary endpoint. You analyze interim data from subjects in the study, not external to the study. It can be blinded or unblinded. It can be with or without formal statistical hypothesis testing. So one example of an adaptive design, it's I don't consider it adaptive, but everybody else does. Changing your inclusion or exclusion criteria mid-study for one of multiple reasons. It could be that you are expecting a certain broad basic characteristics in your patient population and you're not seeing it. And so you're having to modify something in your inclusion-exclusion criteria in order to broaden that base. It could be the fact that you're not enrolling at the speed at which you would like, and so you're going to look through your criteria to determine which ones folks are failing at and ones that you're able to modify in order to improve that success rate. That's what you'll target for adaptation. those are in a blinded fashion and really would have little impact on the statistical validity to most extents. Motivation, more efficient studies, possibly shorter duration, possibly fewer patients. Can use it to drop ineffective arms, doses, or subgroups of patients. You increase the likelihood of success on your study objective by taking a look at the interim data. Do you need to enroll more patients? Do you need to actually target a separate subpopulation in your patient group in order to move forward and progress in a more efficient manner? And you're able to get more information. You're able to look broadly at the beginning, so at a broader set of, let's say, dose information or a broader set of enrollment criteria, and then based off the interim, you're able to focus that criteria. Again, this is all based off of pre-planned modifications, though, so you're going to have to take time up front to lay out what those possible decisions are going to be and understand what the impact has on your statistical rigor. Disadvantages, the key thing here, and you've heard this throughout, is operational bias. I attempt later on to go through and identify some operational biases, and it's really tough to. I think Ken and Joe hit on better ones than I, but I will also target some. Essentially, the bias is an estimate of treatment effect that deviates from its true value based off of something that is going on in the study. Knowing results of various decisions, even if it's only the decision. So if it's a decision based off of endpoints, subjects, sample size, that lead to a change in the conduct or subjective decision making. So for instance, in an area where you're possibly switching the primary endpoints in the middle of the study, now all of a sudden the investigator, if they know that you're switching that, first the question is, do they have to know? But if they know that you're switching that, how does that impact how they're actually taking the measure and recording that measure? There's going to be some bias involved. Inflation of a type 1 error. This is maintained in adaptive design, and that's why the whole prospective planning comes into place. You want to prospectively plan for all the possible decisions that you're going to make to ensure that that type 1 error, or the error of saying, all right, this product is better than the control or non-inferior to the control, that probability stays at a two-sided alpha over 5 in general. And it requires simulations for complex design characteristics. There really aren't programs out there that you can just press a couple buttons and it'll tell you. So there's more effort at the design stage based off of everything I just said. Frequently difficult to interpret if it's an overly complex. So you design. So you really want to target 1 or 2 modifications at most. If you're starting to target 4, 5, 6 modifications, your statistician might have some fun in the short run, but at the end of it, trying to actually interpret the data will be very difficult. And it can also increase the sample size and duration just because of the number of looks in the alpha adjustments. But it also allows you to understand your data midstream. Possible modifications are to study eligibility criteria, the randomization procedure. Do I want to mid-study I take a look at the information and I see I've got a lot more variability in one of the treatment groups than the other. I've known that that's possibly going to be the case, so I've set up a modification to change from a 1-to-1 randomization to a 2-to-1 randomization in that case. Treatment regimens. You can think of basically a dose response situation where you're dropping a dose in the mid-study. If you're not quite sure of what doses to go into your phase 3 with or your pivotal trials with, so you go in with 2 or 3 doses, take a look at the data, again with appropriate designs, and make decisions to drop a dose regimen. Total sample size has been discussed, schedule of patient evaluations, primary endpoints, the order of secondary endpoints. If that order is, a lot of times there's a lot of secondary endpoints, really difficult to order, how you're going to put that up front as far as maintaining alpha and really trying to select that right order. The interim analysis can help with that on an adaptive design, and analytical methods of endpoints. Types of data that can be used, I'm going to go through this pretty quickly. Basically, demographic baseline characteristics, I gave a brief example of that already. Safety outcomes, pharmacodynamic or biomarkers, as well as efficacy outcomes. And again, this can be blinded or unblinded, but the reason I put this up here is, as a statistician, I always get the request of, well, as long as I only call them treatment A and treatment B, that's still blinded, right? It's not blinded. That is unblinded information. So anytime you go to that level of information in order to make a decision, that is considered unblinding. Candidate studies, pretty standard. You've probably heard this before. The key thing is timing of endpoint that you're going to be making decisions based off of relative to enrollment enabling adaptations. So if you've got an endpoint that you're looking at that's 2 years out and you're able to enroll in 2 months, the full study, you're not really a candidate for an adaptive design. Or if you have a shorter term endpoint that's predictive of the primary endpoint, once again, few targeted modifications. So if you're finding yourself in a situation where there are many unknowns, definitely would suggest another phase 2 or early phase trial that's more exploratory to get more information on those unknowns. And then finally, if your samples, this doesn't account for everything, but if your sample size is driven by safety and you're trying to reduce your sample size, then it might not be a meaningful way to use your time for the adaptive, because you have to have the sample size to drive your safety profile anyway. All right. I'm going to go into examples. Am I speaking at a good speed? All right. First example I have is a sample size reassessment study of ADHD, a randomized 1-to-1 test to control. The primary endpoint is a physician-rated ADHD scale. I specifically picked this endpoint because it's very subjective. The considered modifications are sample size reassessment, and that the separate decisions by gender. For some reason, there's a known aspect of this drug that it possibly works different by the 2 genders, and so that the sample size re-estimation decisions are by gender. So this next bullet, the design properties in varying scenarios, I've removed that from the slides from here forward, but please understand that it's there. It's for space reason. For every one of these, we would need to walk through the scenarios that are possible in these modifications, determine decision rules, and then understand from those decision rules expected sample size, expected duration, maintaining the type 1 error and calculating the type 2 error. And in that way, you're able to take a look at the adaptive design that you've set up. You can compare it to conventional design, you compare it to other adaptive designs to see if it's going to give you benefit to run this adaptive design. And that's a lot more difficult than 2 bullets, but that's what we'll go through here today. The key things that I really want to discuss, again, are operational impact and bias. So there's a couple of things here. Let's consider that when you did the sample size re-estimation, you require significantly more females than males. And so I have this next bullet. I didn't quite know where to put it, so I put it there. But let's talk about that on the requiring significantly more females. And as discussed earlier, if that gets back to the investigator or if that gets back to the sponsor, there's possibly going to be some operational bias injected because now they're going to intuit that, all right, this isn't as effective in females. And so what is that going to do to the measures that are taken? And one of the key things that I've been asked to do, and it was brought up in the question and answer, and that is analyze the data pre and post interim to see, all right, is there an effect by stage? Thankfully, again, I as well have not come into a situation where there's been a difference and that I've had to justify, though I will talk about another scenario right after this where I can believe there would be, and so I wouldn't recommend it. So some of the other questions on the operational impact that I get frequently are, should we stop enrollment during the interim or continue on? And that, to me, really depends on what your enrollment rate is, what the cost of the patient is, what the cost of stopping is, and restarting a site, because it's not free to stop and then restart. To get the sites re-energized and going back, it takes a lot of time as well. So that has to be a discussion. And the key thing here for me is the different sites enrolling pre to post interim. I've had this a lot on adaptive designs or where there's an interim for sample size re-estimation, where For some reason, folks will want to focus on their key sites right away. Maybe it's just the first sites that start up. It's their primary sites. They go in, they get those started up. Let's say they're US sites. And then they'll enroll on that, make the decision, while they're opening rest of world. And now rest of world is what's enrolling the next set of patients. And so you can see there that that's going to inherently possibly add bias because we're in two separate areas now of the world. And so I would caution you to strongly think about that as well. And I see it in phase 2 to phase 3s where folks will target their phase 2s on a couple of key investigators, but then want to take that information and develop their phase 3s. And they're not going to have the same variability frequently in those phase 2s because of those investigators that they targeted and because they're targeting only 1 or 2 instead of the 20, 30 that they see in phase 3s. So my next example is a dose pruning, and this is more of an example of some pre-thought to designing the study. And so when people think of adaptive design, they look at the statistician. I, as a statistician, like to look at everybody else because it's not just us. We'll do the mathematical part, but there's a lot of operations that also has to be accounted for, and that's really what I'm trying to discuss here. So if we have an example of 3 active doses and 1 placebo, that can also be 2 active and 1 placebo. The number 3 isn't magic. We just don't know what doses we want to go the whole way through our phase 3 with, but we want a phase 3 now. And so the considered modifications would be to discontinue non-efficacious doses and possibly modify the randomization, which is what I mentioned earlier, from a 1-to-1 to a 3-to-2. And so the operational impact, after doing the impact that I described earlier about looking at the different scenarios, the type 1 error, type 2 error, et cetera, operational impact, are we using an IRT system? Are we set up in that IRT system now for the possible modifications? Because the last thing you want to do is make a modification and now have to go into your randomization system, update it, and retest it all to make sure it's working correctly. Pre-build that into your system. And I've done that, and it works very well. You get to test everything right up front. It really helps from the beginning. So these are just some things to think about. Manufacturing, kit product and availability. You're going to have more call for some of, if they're different kits essentially, or for the different doses, you're going to have more call for the doses that remain on study. Make sure that you've got that available. And then also on the shipping side as well, that, I jumped across resupply algorithms. They would possibly change as well, and then shipping. All right. Here's an example of a primary endpoint. Where I'm looking at a study of dry eye, and I've got hierarchical endpoints where I'm going to test corneal staining first, and if that's significant, I'm going to test the dryness symptom. Both of these are valuable endpoints. I could go multiple different ways here. I could go co-primary, where I'm doing a Bonferroni correction, and I'm going to claim the study is a success if either of those 2 meet. I could go higher, but that's going to reduce the alpha, cause a bit of an increase in sample size. There are other also outside of Bonferroni. I could go a hierarchical endpoint where I'm going to pre-state corneal staining dry and then dryness, but I'm not sure. All right, is that really the way I want to go? The data maybe on earlier phase studies really weren't clear enough, but this was a general direction. I can have an adaptive design set up front where my considered modifications are order of hierarchy and the sample size reassessment. So you're going in and taking a look at that hierarchy at the interim, where you might have a lot more information than you've had from your prior studies, make a decision and move forward. Operational bias here, concerns. If, all right, all of a sudden you were, going corneal staining as primary, and then dry eye or the dryness symptom is secondary, and now you switch if the investigators know about that or others. What is the impact that that's going to have on their assessments of these endpoints? I can't tell you. That's the concern. So must the investigator know at this point that this decision has been made to switch? And I would argue they do not need to know in order to basically maintain status quo and to not interject operational bias. You can make that decision and not pass that forward to the investigators. It's not meaningful to them in their assessments. All right. Okay, and then lastly on the composite endpoint, I was trying to come up with a couple of examples for different areas. This one is really to ensure collection of the data on the EDC system up front, that you account for any possible modification in the study based off of an adaptive design, what impact that might have on the EDC system. I also threw in here risk-based monitoring, so let's walk through it. So if we consider our primary endpoint as a composite endpoint, but we have an adaptive design that actually allows us to change what assessments go into that composition. Pre-stated, whatever they are, we do the assessment of alpha, power on the stat side. Operational impact questions, I'm assuming all the measures that are possibly going to be modified to in the composite are already collected in the EDC. You need to have that to assess whether or not it makes more sense to change out those individual assessments in the composition. What about the risk-based or targeted monitoring impact on this, possibly on an adaptive design? I've seen some areas where targeted monitoring, they're focused on the primary endpoints. That's what they're going in and source data verifying, as well as a random assessment of other things. Now you're going to, midway through the study, change what's possibly going into that composite endpoint. So it's something to think about with respect to how do you build that system in order to change over midstream, because you don't want to be doing those types of changes mid-study. Pre-build that. So this is, I'm really trying to understand, basically point out that while this is statistics heavy, it also has a lot of operational impact. And then operational bias here, of course, pretty similar to what I've been discussing. If investigators are knowing that you're pulling out a certain assessment from the composite, then they're probably going to be thinking, all right, we're really not seeing efficacy there, but we are really seeing efficacy on these other assessments. That's why they're putting them in. What does that do to their assessing of those endpoints? This is my last slide. Sorry if I went. This is the seamless phase 2-3. This is, I just put this on because this is what everybody frequently thinks about on adaptation. And I would just really caution that, a lot of times when this is asked for, there's more than one or two modifications that are being thought about. There's a lot of not understood information about the product or the product in the indication. So you really just have to weigh, are there really only a few things that I want to understand and possibly modify versus do I need to run a phase 2? Take the time to truly analyze that data from a safety perspective and an efficacy subgroup to get a full understanding of that data. That's it. Thank you.
I actually wanted to understand, I guess you had a very good examples about changing the primary endpoint, right? So there is a big regulatory risk by changing the, if you go to the FDA, talk to them, they hate it, right? Because we have been there several times about it, and first of all, so I'm just wondering, how did your regulatory interactions go, number one, and we have not seen a single scenario where they have approved changing the combination of a primary endpoint, because there is an inherent question about how are you going to do your predictive power, how are you planning to do your conditional power, and if you're trying to make a decision based on a primary endpoint on a conditional power based on one endpoint, what's the second endpoint looking like? Especially if it's a pivotal trial, it comes to a point that the pivotal endpoint is actually regulatory approved endpoint. So how are, so I'm just very interested to know about the regulatory interactions, because my experience is we have horribly failed.
Yeah. So they're definitely challenging, for sure. And I think I've possibly been in scenarios where the primary endpoint hasn't been as well defined, and they are, because of that, a little bit more open to hearing the adjustment. The composite, I have not done this one. This is an example from one of the guidances. Well, I created the example, but the composite endpoint itself was an example from the device guidance straight up.
We're talking about adaptive design and then possible changes. And I think what's throwing me in my head is that in my mind, when I think you're running an adaptive trial, you're thinking ahead of what you could do. Just because you make a change, whether it be a result of a DMC oversight or whatever, does that automatically make it an adaptive trial? Like, I'm a little confused there. It's like almost like we're mixing the terms.
Okay, I apologize. I'm definitely missing your question.
Okay.
So the pre-planning on the adaptive design is key in order to really understand the working characteristics of the underlying statistics. So are you asking?
Just making a change in your study doesn't make it an adaptive trial. It's showing that you've made a change.
And depending on what that change is and what information you had when making that change could make it a study that's possibly not worth continuing because it's not going to be looked at in a positive fashion. So
but, I mean, you had said earlier that you didn't think a change in inclusion and exclusion criteria was an adaptive. And I agree. I mean, to me, it's showing you that there's some parameter of the conduct of your trial or your criteria that you need to change. But in my mind, when we say we're going to have an adaptive trial, your protocol is going to state, after enrolling the first 100 patients, if we don't see certain criteria met, we will, lessen it or, something like that.
No, so I am in complete agreement, and that's actually why I threw out that example and said I would not have recognized it as such. The guidances consider that adaptive. So
you're adapting to whatever is.
Sorry.
No, you're adapting to either the safety profile, the efficacy profile, or operational profile.
Exactly it. So it doesn't have to be on efficacy data, as I had in one place. It doesn't have to have any formal stats. You're basically taking a look at the information in the study, so it has subjects in the study, and making a change in that with that information. Yeah, absolutely.
Can you comment on the advantage versus disadvantage ratio for running an adaptive design for a superiority trial, let's say versus placebo, versus a non-inferiority? Is there anything in these two basic structures that say, better adaptive or not, based on whether it's in a non-inferiority or a superiority trial, all other things being equal?
Yeah, that's a really good question, because I do that in pretty much everything else I do in statistics, and I haven't thought about it in this situation. Yeah, I think the only thing I would possibly caution against or think to in that situation is that operational biases that might somehow lead to overstating efficacy. I would have to look at how the operational biases impact the primary endpoint with respect to, are you going to be more likely to claim non-inferiority because of the operational biases, or less likely, and how that compares to superiority. And I will think about that and get back to you. I apologize, I do not have an answer.
Thanks.
Yeah.
Thanks for your talk. I enjoyed it. Could you give us an example of where the seamless phase 2-3, where you would recommend it and where it would have clear advantages?
Yeah, I personally, so if you have a lot of information on the product, so I would say in general, I personally don't recommend it because I think people try to do it as a way to shorten their time, and what ends up happening is while they find out some information on the phase 2, they are more likely to actually have to change the phase 3 aspect of it mid-study. So if the product is well known, your serious adverse events, your safety profile is well known, and possibly a similar indication, you also have good information on the efficacy with the indication that you're talking, and you're just maybe looking at a focused set of dose ranges in the phase 2 from what you've done previously, just to make a decision on what dose ranges to move forward with in the phase 3. So I would recommend it in scenarios where you have a good amount of information on the product, possibly in the indication, and also have only a couple of unanswered questions.
Dale, when you mentioned statistical analysis, is it possible to take an adaptive design where you check to see whether there's baseline imbalances between treatment groups and then change the analysis to a change from baseline, or or an regression analysis with the covariates. Are those acceptable?
So technically, per the guidance, they do mention similar to that, though, as mentioned previously on another, I've not personally been successful in this situation where I'm looking at an interim information and then basically designing it to adjust for covariates based off of that. But that might be division specific as well.
All right, thank you, Dale.
Thank you very much, presenters. Thank you so much. Thank you all as well for joining us. And have a great evening and safe ride home.